Bismillah الرحمن الرحيم. Today we will talk about red blood cells morphology presented by me, Dr. Amira Hosny, Master of Clinical Pathology in Shams University. In this lecture we will talk about four main things about the red blood cells: the normal morphology of red blood cells, the poikilocytosis, which means differences in shape, inclusion bodies in red blood cells, and we will illustrate some few cases about red blood cells morphology. First, normal red blood cells. Before discussing the normal shape of red blood cells, we will start to explain the process which is called hematopoiesis. Hematopoiesis is synthesis of blood cells. Here, if we talk about the main cells of the bone marrow, it is the hematopoietic stem cells. The hematopoietic stem cells will differentiate into multipotential stem cells, which will give two main lineage. This two main lineage, one of them is called the lymphoid progenitor lineage or lymphoid lineage. This lymphoid lineage will be differentiated into natural killer cell, T lymphocyte and B lymphocyte. And this myeloid lineage will be divided or subdivided into granulocytic lineage that includes neutrophil, basophil, eosinophil, monocytes, which will give the tissue macrophage, and another lineage is called the megakaryocytes that will develop or that will give platelets, and another lineage is called erythroid lineage that will give red blood cells. So mature red blood cells come from erythroid lineage, from myeloid progenitor cells, from multipotential stem cells, which was hematopoietic stem. This illustration is more clear if we say that we start with multipotential hematopoietic stem cells. It will give us common myeloid progenitor that will give us the mature erythrocyte. The first cell to appear in the bone marrow is called the erythroblast. What is the description of the erythroblast? It is called the basophilic erythroblast or normoblast that has a diameter smaller than the pro-erythroblast. The nuclear chromatin shows a strong contrast between the light-colored areas and the dark violet chromatin particles. The cytoplasm is moderately basophilic, reflecting high protein and RNA contents. So if I check this cell, this is called erythroblast. This is the second cell after the pro-erythroblast. If I check this cell, I will find that here there is deeply basophilic cytoplasm, and here there is condensed mature nucleus with a stippled appearance. What does a stippled appearance mean? Means that there is very compact chromatin here and some clear areas, completely clear areas in between this nucleus. This is the maturation of the erythroid lineage. This is the pro-normoblast, early normoblast, intermediate normoblast, late normoblast, and mature red blood cells. If we talk about this cell, it is called normoblast. What is the normoblast like? This normoblast is larger than the normal red blood cells, as we see in this photo, and it is nucleated like this. The nucleus has very dense chromatin, and it is slightly pycnotic like what we see because it will be extruded outside the RBCs, and it is surrounded by cytoplasm. Actually, this cytoplasm is the hemoglobin of red blood cells and almost without any inclusions as what we see now. So this is called normoblast. Normally, this normoblast is an immature red blood cell which is not present in peripheral blood. It should be present only in bone marrow. So if I saw this normoblast in peripheral blood, it comes with many conditions like hemolysis or hemolytic anemia or bone marrow infiltration. Sometimes we see it in leukoerythroblastic reaction. This is the same cell, normoblast. Here in this slide, I have another nucleus of another cell. Why this nucleus? This nucleus is slightly larger than this nucleus in this cell. This is the nucleus of lymphocyte. I bring it here just to compare the shape of lymphocyte with the shape of normoblast, because sometimes it is overlapping together. The nucleus of the nucleus of lymphocyte is of the size of red blood cells, like 1 to 1.5 times. But the nucleus of normoblast is a small nucleus because it can enter into the red blood cells. So if you check this nucleus comes into this cell, but this nucleus cannot, and it is surrounded by very rim of cytoplasm. So this is the difference between normoblast and lymphocyte. Well, we have another cell that is called polychromatic cells, and we see in the peripheral blood film. What is the polychromatic cells? If we said that this one and this one both are polychromatic cells, what is the difference between this cell and this cell or this cell? If we say this is the normal red blood cells, what is the difference between this and this? This cell is slightly larger than the normal red blood cells, larger by about 20 to 25%. And there is another difference, that here there is some central hypochromia, but here there is no any hypochromia. Here it is fully hemoglobinized, that make it slightly more darker or slightly darker than this cell. So this cell is larger and darker. What is this cell called? It is called polychromatic cells. This cell is very significant for many diseases, and to discover the actual shape of this cell we have to do another stain. The other stain is called supravital stain. This supravital stain represents what is called reticulocytes or retics. What are the reticulocytes? The reticulocytes are the premature red blood cells, that they are the cells that we saw in the previous slide. Premature red blood cells which are slightly larger by 20% and containing some few blue dots, as what we see with the supravital stain, like brilliant chrysal blue. These blue dots are the remnant ribosomal RNA which appears in this retics. Normally we have retics percentage like from 0.5 to 2.5%. But if there is reticulocytosis or there is increase in the reticulocyte production index, this means something is going on, like hyperactive bone marrow in case of hemolysis, hemolytic anemia. But if we see that retics count is depressed or retics production index is less than 2, this occurs in case of ineffective erythropoiesis, like in case of myelofibrosis, in case of myelodysplastic syndrome, and like megaloblastic anemia. This is another slide for reticulocytes. Here I'd like to compare the shape of premature red blood cells in Wright stain, James stain, or Leishman stain, that they are larger and darker cells, like this one and I guess this one also. If I check these cells with brilliant chrysal blue, which is called supravital stain because it stains vital cells or leaf cells, we will find that this reticulocytes containing these blue dots, which are, like what we said, remnant of ribosomal RNA. So finally we reached the shape of normal red blood cells. As what we see, red blood cells normally is discoid in shape, and that helps it to be more flexible and deformable, and it shows central hypochromia, like what we see here. Now we finished the normal maturation of red blood cells from the beginning of pro-normoblast, passing to normoblast with all its different stages, and also the premature RBC, which is called the Normoblast, and the Reticulocytes, and finally we reached the mature red blood cells. Now we will talk about the poikilocytosis. What is poikilocytosis? Poikilo means difference in shape, and anisocytosis means different in size. Here we will focus on poikilocytosis. Let's go. The first slide is what is called ovalocyte. What is this ovalocyte? Ovalocyte is what we see now. The RBCs are not longer like this rounded red blood cells. It is now like oval shaped ovalocyte. We can see this ovalocytes in many diseases like hereditary ovalocytosis, like sometimes in thalassemia major, and sometimes also in megaloblastic anemia. And now this cell is called elliptocyte. What is the elliptocyte? It is slightly looks like ovalocyte but is more compressed. Sometimes we call it pencil shaped cells, like the pencil. And it occurs in hereditary, we can see it in hereditary elliptocytosis and in iron deficiency anemia. This one is called stomatocytes. This is stomatocytes, like the red blood cells containing few slits inside, and this is stomatocytes we can see in alcoholism, liver disease, and hereditary stomatocytosis. And let's go to another cell. This one is called a teardrop cell. Check, this one is like the teardrop. This teardrop cell is commonly seen in what's called the myelofibrosis. We will discuss later in white BC's morphology and white BC's abnormality, myelofibrosis. Also we can see in megaloblastic anemia, and sometimes we see it in iron deficiency anemia. Now we have another cell which is called spherocytes. This is spherocytes. What is the difference between this cell and the normal red blood cells? It is confusing sometimes. Yes, it is confusing, but there is many other methods to diagnose if there is spherocytosis or not. This spherocytes is very regular, very rounded red blood cells with absence of central pallor here. Regular, rounded, with absence of central pallor here. And this spherocytes occurs in many conditions, hereditary and acquired. Hereditary, like hereditary spherocytosis, acquired like immune hemolytic anemia and hemolytic disease of the newborn. Now we will transfer to another cell, which is the target cell. What did the target cells looks like? This is the shape of target cell. Rounded cell with central condensed chromatin. It is characterized by central darker area surrounded by lighter zone surrounded by another darker zone. This is the target cell. Where can I see the target cell, or what are the conditions that we can see target cell? In the peripheral blood, we can see in thalassemia and hemoglobin disorders. We can see also in liver disorders like chronic liver disease and in iron deficiency anemia. Now another cell called sickle cell. This sickle cells looks like the shape, sickle shape. Sickle cells occurs in sickle cell anemia, which has many types, like sickle cell disease, two main types, like sickle cell disease, the hemoglobin is called hemoglobin SS, and sickle cell trait, the hemoglobin is called hemoglobin AS. In this film we can see sickle cell and we can see also target cell. This might be sickle cell disease or might be sickle thalassemia. How can I differentiate between two diseases looks like each other in the blood film? I can differentiate by very simple technique, which is called hemoglobin electrophoresis. If in the hemoglobin electrophoresis I found all the band or most of the band in the region of S region, so it is almost hemoglobin S disease, which is sickle cell disease. If I found that there is band in S region and band in A2 region, band in F region, this can be most appropriately sickle thalassemia. Another cell which is called schistocytes. What are these schistocytes? Schistocytes are called fragmented red blood cells. Fragmented red blood cells means that there is something intravascular, this is very important, something intravascular that leads to destruction of this red blood cells. Like what? Like thrombosis, like severe vasculitis. This occurs in many conditions like non-immune acquired hemolytic anemia, for example in case of microangiopathic hemolytic anemia like TTB, thrombocytopenic purpura, hemolytic uremic syndrome, DIC, and other conditions like PNH, paroxysmal nocturnal hemoglobinuria. All these conditions, there is intravascular hemolysis leads to fragmentation of red blood cells, give this shape which is called schistocytes, like this one and this one. Well, another cell which is called acanthocytes. What is this acanthocytes? It is red blood cells with peripheral projections. This acanthocyte we can see in liver disease and sometimes in post-splenectomy. Now we have another group of cells which are called Roleux formation. In this Roleux formation, RBCs like here, like touching to each other like stack or aggregates, but in a very unique shape because of the flat surface or discoid surface of the red blood cells that makes it give this picture. Roleux formation, sometimes we see it normally, but sometimes we can see it in other diseases like multiple myeloma, Waldenstrom macroglobulinemia, sometimes in inflammatory disorders and sometimes in autoimmune disease. This might be due to excess antibodies, that antibodies make red blood cells stick to each other. But in this film we will see what is called agglutination. What is this agglutination? It means that red blood cells making groups, clusters, huge groups like this group and this group. There is big difference between this one and the previous one. The previous one was like homogeneously in shape or unique in shape like line of red blood cells, but this like groups, like clumps of red blood cells. This autoagglutination occurs in cold agglutinins and warm agglutinins like autoimmune hemolytic anemia. Now we finished the red blood cells poikilocytosis, and now we will start another topic which is called red blood cells inclusion. Let's see what are the red blood cells that we have today. First, Howell-jolly body. Howell-jolly body is like small single blue dot intracellular inside the red blood cell. What it expresses? It expresses remnant of nucleus, remnants of DNA or remnant of nucleus, and this occurs in case of megaloblastic anemia and also in post-splenectomy. Sometimes we can see in other types of hemolytic anemia, but it is really characteristic of megaloblastic anemia. This one is called Cabot ring. If we talk about Cabot ring, we will see that this is the red blood cell and this is the Cabot ring. What is the Cabot ring? Cabot ring is the remnant of the nucleus also. The remnant of the nucleus, and this also occurs in megaloblastic anemia. This Heinz body, as we see, it occurs in some condition like G6PD deficiency, like oxidant drug, and some cases of hemolytic anemia like alpha thalassemia. Now we have another slide for another inclusion called siderotic granules. What are these siderotic granules? Siderotic granules are granules of hemosiderin. See, this is blue dot, but here each cell containing many blue dots, not only one single blue dot like what we saw in megaloblastic anemia, but here I have, for example, in this cell, two blue dots, here two, here two, here two, here three blue dots. This is called siderotic granules. Siderotic granules like excess hemosiderin. So hemosiderin is the storage form of iron. Iron has two storage forms, ferritin and hemosiderin. The stainable form is the hemosiderin that can be stained with the iron stain, and we can see this hemosiderin granules. This hemosiderin granules occurs in cases of excess iron. Okay, cases of excess iron like hemolytic anemia, like post-splenectomy. Sometimes we see in other condition, as what we will discuss later. Here we will talk about basophilic stippling. This is basophilic stippling. This occurs in case of absence of 5-nucleotidase enzyme. There is remnant of RNA, many remnant here as what we see, and this occurs in case of lead poisoning, heavy metal poisoning like lead, sometimes in hemoglobinopathies like thalassemia, but is not that common. It is common in lead poisoning. And now we have hemoglobin H inclusions like what we see here by brilliant grisaille blue. This is called hemoglobin H inclusion. Hemoglobin H, or hemoglobin parts or parts hemoglobin, which means deletion of 3 alpha chains. Well, now we will just enumerate few cases, and we will interpret the different morphology that we saw now and the different inclusions that we saw now with different cases of red blood cells that we see frequently in our work. First case. This blood film shows many different cells. Like if I check this cell is pencil shaped cells, and this cell is hypochromic microcytic cell. This one is also microcytic hypochromic microcytic, and some small target cells, and here teardrop cells, and this is our small platelet. Here there is what is called anisopoikilocytosis. Anisopoikilocytosis means different in size and shape, and this occurs in common syndrome which is called iron deficiency anemia, common disease or common syndrome called iron deficiency anemia. Iron deficiency anemia is the commonest anemia, and the most common cause of iron deficiency anemia is bleeding in case of occult blood, excess menstruation, and many other causes leads to iron deficiency anemia. Sometimes it occurs due to defective intake in the iron or increase in needs of iron like adolescence, like pregnancy, or bleeding as what we said, all defect in absorption of iron like hypochlorhydra or something like that. Its treatment is very simple like iron, and regarding the diagnosis of iron deficiency anemia, it depends on many factors from the clinical picture of the patient and the CBC. Here regarding the CBC of the patient, this patient will have anemia and with microcytosis and hypochromia of the red blood cells, and this patient will, if you read the indices of red blood cells, you will find that there is decrease in the MCV and MCH, and there is increase in the red RDW, which is called the red cell distribution width. Sometimes the red cell distribution width is the first parameter or the first one of the indices to be changed in the CBC, because in the early stages there is some cells will be hypochromic and other cells are still normochromic cells, so the red cell distribution width will be changed. Well, we will transfer to the second case. What is this? If I said this is the iron stain of the bone marrow, and this is nucleated red blood cells. What is the nucleated blood cells we call? We call normoblast, as what we said. Here we have two different normoblasts. One looks like this, showing few siderotic granules around the nucleus, and the other one like this, show other siderotic granules but around the nucleus, like collar shape surrounding the nucleus. What is the difference between this one and this one? This one is called normal sideroblast or siderocyte. Normal, like there is some few siderotic granules scattered intracellular here, but in this case the siderotic granules are surrounding the nucleus. Actually they are surrounding the chromatin. Why? Because to build up our hemoglobin we need to have heme and globin. Heme is formed of iron and protoporphyrin. Here there is iron, we have excess iron, we have many iron absorption, but to form heme we have to have protoporphyrin. Here there is absence or severe deficiency of protoporphyrin, so there will be failure of conjugation of iron with protoporphyrin due to deficiency of protoporphyrin. So this results that iron is surrounding the mitochondria because it cannot enter inside to be conjugated with protoporphyrin because there is deficiency already in the protoporphyrin. This occurs in many conditions, or this have many causes, like hereditary sideroblastic anemia occurs in case of deficiency of allosynthetase enzyme, and also in acquired sideroblastic anemia. Sometimes occur in myelodysplastic syndrome, specific stage which is called RARS, refractory anemia with ringed sideroblast. In this condition the ringed sideroblast reflects more than or equal 15% of bone marrow normoblast. Here there is another picture of the blood film which is called dimorphism. Why I'm talking about dimorphism just after the sideroblastic anemia? Because dimorphism is highly characteristic of sideroblastic anemia. Here in this slide I can see two different population of red blood cells showing central hypochromia, not only central, showing marked hypochromia in the RBCs, and another population fully hemoglobinized. This is called dimorphism. It is characteristic of sideroblastic anemia, but there are other conditions characteristic with dimorphism, like iron deficiency anemia on treatment or with blood transfusion, or any condition with blood transfusion, like megaloblastic anemia also with treatment or with blood transfusion. Well, now we will be transferred to another case. If I check this is a film of bone marrow, this is bone marrow film, and this is the erythroid series. If I check this cell, this is the erythroid series. There is some differences of this erythroid series with the pro-normoblast and early normoblast that what we checked before. If I checked here, I will find that there is delay in the maturation of the nucleus. There is delay in the maturation of the nucleus. That what we call the nuclear maturation lags behind cytoplasmic maturation. So, megaloblastic anemia, there is deficiency of B12 and folic acid. Both of them are very important for DNA building, DNA maturation, means nuclear maturation. And we said before that in nuclear maturation in the erythroid series, the nucleus becomes condensed, smaller, then it is extruded outside the cell. If there is deficiency in B12 and folic acid, mean there will be delay in the maturation of the nucleus. So we will see some megaloblastic changes. This megaloblastic changes mean large nucleus or nucleus cytoplasmic asynchrony. Sometimes we can see nuclear lopulation, nuclear fragmentation, nuclear segmentation, and sometimes also we can see mitotic figures of the erythroid series. This picture we call megaloblastic features that we can see in megaloblastic anemia that have many causes. Here the erythroblast, or we can call it megaloblast. Check this one. We find here that very large nucleus, and this large nucleus showing many stippling inside. Also like this one, very large nucleus and many stippling inside, stippled appearance. This also the megaloblastoid features which occurs in the megaloblastic. Other specific feature for megaloblastic anemia that commonly we can see in the peripheral blood in case of megaloblastic anemia, what is called hypersegmented neutrophil. As what we see here, this neutrophil has many segments, so I just would like to know what are the normal segments of the neutrophil nucleus. The normal segment is from 3 to 5 segments. Here I find like 1, 2, 3, 4, 5, 6, 7, 8 segments. Each segment means it is a hypersegmented neutrophil. This hypersegmented neutrophil occurs in case of megaloblastic anemia. Well, I'll transfer to another case, but I will not discuss this case. I just bring the shape of the cells to compare with the erythroblast. This cell is called plasma cell, and this plasma cell occurs in plasma cell dyscrasia or plasma cell disorder. One of the commonest is called multiple myeloma. Sometimes it is confusing with the erythroblast, but if I checked here the difference between plasma cell and the erythroblast, the plasma cell looks like oval cell with eccentric cartwheel nucleus, and here there is halo around the nucleus or surrounds the nucleus, which is called perinuclear halo. This represents Golgi apparatus, which is hardly working, hyperactive due to synthesis and release of antibodies. Like this one is very clear, like the difference between plasma cell and erythroid cell. Here we have also another shape for the plasma cells, or another slide for the plasma cell, oval cell with eccentric nucleus surrounded by perinuclear halo and deeply basophilic cytoplasm. Well, now we will be transferred to another case which is called spherocytosis. Check this shape of the red blood cells. As what we said before, it is rounded, strictly rounded, regular cells, fully hemoglobinized inside, like this one, this one, this one. This is called spherocytosis, okay, or spherocytes. As what we said before, spherocyte can be hereditary spherocytosis. This is one of the hereditary hemolytic anemia, causes of the hereditary hemolytic anemia, and it can be in case of autoimmune hemolytic anemia. There are other tests to diagnose spherocytosis, like osmotic fragility test. Well, there is a picture for osmotic fragility test. This test expresses the ability of the red blood cells to tolerate hypotonic saline. Here in this photo we have serial dilution of saline. We started with 1%, 0.85, 0.66, like this, and here is the same serial dilution, and here is the control, and here the suspected patient. If I check here, I will find the hemolysis started almost from 0.66, but the manifest hemolysis here from 0.38. This is because the red blood cells normally are discoid shape, slightly flat, have some space inside. So if I incubated the red blood cells with the hypotonic saline, it can take some of this saline inside and can swallow. But in this condition, in case of spherocytosis, notice that the red blood cells are very rounded, are very rigid, that if the red blood cell swallows some saline, it can rapidly lyse. Okay, so here hemolysis starts from 0.85 and we can see manifest hemolysis from 0.58 or even less. Well, another case called G6PD deficiency. Before we talk about the morphology of cases, I'll just remind you with the G6PD enzyme. G6PD enzyme is very important in glucose pentose shunt, which will provide glucose 6-phosphate dehydrogenase. Glucose 6-phosphate dehydrogenase enzyme will provide NADPH, and this NADPH is very important for glutathione reductase. Glutathione reductase is an important antioxidant. In case of absence of G6PD, there will be deficiency of NADPH and deficiency of glutathione reductase enzyme. And if this patient with G6PD deficiency is exposed to stress like hypoxia, like some excess oxygen free radicals, or some drugs like aspirin, like cephalosporins, like anti-malarial drugs, this will lead to consumption of glutathione reductase enzyme. This consumption of glutathione reductase enzyme leads to sudden shift of the hemoglobin into one side of the red blood cells, leads to what we see here, that's called basket cell. This is called the basket cell, as what we see as if the hemoglobin is shifted to one side and one side of the red blood cell is empty. Another slide will illustrate it more. For example, here, if I check this cell, there is here rim of cytoplasm or rim of the red blood cell membrane, but all the hemoglobin is shifted to one side. Also in this cell, rim of the membrane, hemoglobin is shifted to one side. Rim of the membrane, hemoglobin is shifted to one side. This occurs in case of acute G6PD deficiency, what we call favism. Here is the shunt. We just provide that glucose 6-phosphate. In presence of glucose 6-phosphate dehydrogenase, glucose 6-phosphate will be transferred into 6-phosphoglucoronate that provide the presence of NADPH, and this NADPH is very important for glutathione reductase, that is very potent antioxidant. We have another case, I think this is the last one, which is called thalassemia. About thalassemia, this is the peripheral blood film of thalassemia. Here I found some microcytic hypochromic cells, and here I found some ovalocytes, and I found many normoblast. Presence of normoblast in case of thalassemia is a very common feature that we see, because this means there is compensation of this hemolysis, like the bone marrow leads to formation of more red blood cells, and some of these premature red blood cells, which are called normoblast, are released from the bone marrow to the peripheral blood. This gives false high total leukocytic count that needs from us to correct this count, because these red blood cells are nucleated, so they are counted on the culture or in the red blood cell counter or in the counter like white BCs, because they are nucleated. So we need to do blood film and we need to count this normoblast and to do what is called the corrected total leukocytic count to calculate the actual total leukocytic count of this case. So we have here some anisopoikilocytosis. There is difference in shape, there is difference in size of red blood cells, and we have also many normoplastemia. This occurs in thalassemia. Another shape of cells, like what we said before, that's the target cell that we can see also in thalassemia. And finally, I wish you enjoyed my presentation. Thank you. Goodbye.